Hi there, my name is Carlee Stoyanovich and I work in the Porter Lab, a molecular biology lab that focuses on cell cycle regulation. First, let's begin with a quick overview of the cell cycle. Our bodies are made up of trillions of cells that are constantly dividing. This cell division is under the control of checkpoints. When conditions in the cell are favorable, protein complexes will bind at these checkpoints and alert the cell it may begin to divide. Alternatively, when conditions are unfavorable, such as when there is DNA damage, these protein complexes and checkpoints are inhibited, which will cause the cell division to halt. These protein complexes and checkpoints are extremely important and very tightly regulated. It is when the cell division becomes unregulated that serious proliferative diseases such as cancer may result. SPY1 is a protein that has the ability to bind at these checkpoints and speed up the cell division. It can also allow damaged DNA to replicate by overriding the DNA damage response. My project focuses on SPY1 in the liver, one of the most interesting and vital organs in the body. It detoxifies our blood, helps break down nutrients, stores and releases glucose, excretes bile to break down fat, and can regenerate from 1/2 its original size. It is also susceptible to hepatocellular carcinoma, also known as HCC, the most prevalent and aggressive form of primary liver cancer. Current therapies for HCC are invasive and include transplantation, resection, ablation, and chemotherapy. Reoccurrence of HCC with these therapies is common, thus giving the patient a poor prognosis. Many predisposing conditions may make the liver vulnerable to HCC development. Some factors include hepatitis, alcoholism, non-alcoholic fatty liver disease, and non-alcoholic steatohepatitis, which all lead to a fibrotic and/or cirrhotic liver. This compromised diseased liver may then progress into HCC. It has been observed in our lab that mice with higher levels of SPY1 develop increased rates of HCC and fat accumulation. I have been tasked with trying to determine whether these increases are due to the liver injury or are actually caused by SPY1 itself. To establish this causal relationship, I have initiated the predisposing condition non-alcoholic steatohepatitis in wild-type mice using a methionine-choline deficient diet. The liver responds to this damage by invoking its damage response, which includes apoptosis of the cells, fat accumulation, inflammation, and the laydown of fibrotic deposits. Liver tissue was collected at days 2, 7, and 42. So we had long-term and short-term time points to compare. Liver tissue was collected at days 2, 7, and 42. So we have short-term and long-term time points to compare. To verify if the extent of this damage was due to SPY1, I have quantified the SPY1 protein levels in these collected livers and also examined fat accumulation. Future work will include collecting more mice at different time points to assure we have not missed any peaks in SPY1 expression. Apoptosis, inflammation, and fibrosis will all be examined and correlated to these SPY1 levels as well. It is my hope that a new molecular therapy target will be found throughout this research. At the very least, it will aid in understanding the disease progression and characterize the molecular changes.